Okay, just a few housekeeping items before we get started. Hopefully you saw my announcement. By request, I sent you the slides for today's lecture. I said before that I normally don't do that because most of the slides that I have don't have a ton of text. But today's a bit of an exception. So just so you don't have to worry about scrambling to write everything down, you can see what is already on the slide, and if you need to make notes on top of that, just to kind of supplement the content on the slide based on what I emphasize or examples that I give, because those kinds of things are relevant for the term test. So if I give a specific example that's not actually written on a particular slide, it's fair game in terms of possibly being on a term test. So again, whenever the lectures will have kind of heavy content like that, I will post them. Next week, we have a guest lecture from Dr. Handley, and his slides also are a little text heavy, but he will provide the slides to me beforehand, and I will be able to post them before that as well. I didn't realize that the slides that I had last week were not visible to you. No one told me. Just when I came to post today's, I saw that they weren't there. But I know that I sent them as an attachment. So today I purposely didn't send them as an attachment so that I can make sure that you log into the Quarkus page for the course and make sure that you know just exactly where these slides are posted. And they're posted under modules. Okay? The other thing is, and again, I thought maybe we can have this done by today, but it's just going to take a few more days. So in terms of finalizing the groups, so just to make sure here that everyone got the email. So I sent out 2 announcements. Did anyone not get an email from the course today? Not get it. All right. Just to make sure, because in previous years, occasionally, on rare instances, sometimes they get into people's spam or junk mail. So I actually check my own spam almost every day, and almost every day there's a legitimate email that shouldn't have been there. It's a little bit of waste of time, unfortunately, but anyways, just make sure that you're getting the notifications. Another just kind of safe bet is just to kind of check the course page every once in a while and look for announcements and postings. In terms of the sample report from Nutrigenomics, again, you can see the slides from last week in terms of instructions. It's also in the syllabus. If you want to get a copy so you can see what a sample report looks like, that's going to inform your final written assignment. And so for the group presentations, if it's possible that I'm able to finalize it over email, I will do that and send a notification for everyone. Some of you might already know your fellow group members because you asked to be put in a group of 4, I say maximum of 5? I said maximum of 5, right? Yeah, because I think there's 40 of you here. up to 40, and so we have 8 groups. And so at that time, I'll also assign the paper that your group is responsible for, so that you can, get started on that. There's no better time than the present. And also just to kind of split up the work, right? Because remember, up until like the last day to drop a course, it is possible that a group member might drop the course. So you always got to make sure that you have, a kind of a backup plan. If one group member is assigned to, let's say, critique the methods of the paper, and all of a sudden that student isn't appearing, they're not, carrying their weight, or all of a sudden they drop the course, you have to make sure that you plan for that. So obviously, if there are any issues in terms of meeting expectations from the group, and if there is any member of the group that you feel is not carrying their weight, it's happened on occasion, every couple of years maybe, there'll be some grumblings in a group. Oftentimes, I meet with the group and things are sorted out. Sometimes other things happen. Sometimes someone has something serious going on in their life and they just can't do what they're assigned to, but it's obviously not fair to the other members. So definitely, if you feel that there are any issues, just make sure that you're communicating with your group members. There's full transparency and no surprises. The midterm, as I mentioned, the room has not yet been assigned. It's probably going to be in one of those rooms on McCall Street. I'll make sure that you know well in advance. Has anyone had any difficulty accessing the assigned readings? Accessing the book? No difficulties? Has anyone accessed the book? Yeah? Okay. All right. Like I said, it's not meant to necessarily coincide with the lectures. It's just those 2 chapters are covering material that's going to be covered on the midterm. So, you pace yourselves. I've tried to kind of separate them in a way that tries to set a pace for you, but again, it's just up to you how you want to do it. Any questions so far about anything that we've talked about or anything from last week's lecture? So it's a little bit noisy here near Tim Hortons. So, as you can see from the slides today, a lot of this is really kind of, some basic review of genetics, genomics, proteomics, metabolomics. Some of you may have taken other courses in molecular genetics or human biology, and a lot of this is kind of just a kind of a total review for you, but I thought it's important just to kind of make sure that we spend a little bit of time covering these basic concepts, because a lot of these are going to pop up in the papers that you're going to be presenting, and also in some of your assignments. So as we know, most human cells, not all of them, have 46 chromosomes, 23 pairs, and the sequences result in genes. But there's also sections, obviously, that are non-coding. But really, a lot of what we're interested in are the genetic variations in sequences that are within genes or very close to genes, because we're learning more and more every year that a lot of these alterations that happen just outside of a known gene have huge biological effects. So we used to think that our genome was like 95% junk DNA. We actually called it junk DNA as though we didn't need it. Well, we know that's absolutely not true anymore. We still don't know what the vast majority of it does, but gaining more insights as time goes on. So the entire genome for a particular living organism is really what's made up of all these genetic sequences. And the nucleotides are the basic building blocks, the letters, the A, C, G, T. These are the nucleotides that make up the human genome. And as you know, adenine, links up with thymine and guanine with cytosine in terms of the base pairs, the complementary sequences. And so one thing to note, either in a paper that you're reviewing, is sometimes the complementary sequence is what's reported. So not the actual coding strand, but the complementary sequence, meaning the complete opposite. There's been some efforts to try to standardize this so that there's no confusion, but in some of the earlier genetic testing services that were available, including 23andMe, they actually reported in some instances the complementary strand, so rather than the coding sequence. So you can imagine if a polymorphism causes a C to G substitution, you're reporting the G as having a higher risk, but it's actually the C, because you reported the complementary sequence, you're going to get the complete opposite result. And this has happened sometimes, even in publications where authors have had to submit not quite a retraction, but just a correction to their paper saying, oops, we reported the complementary sequence, the non-coding strand, when it should be the other strand. So when we think of common polymorphisms, I mentioned last week that SNPs, single nucleotide polymorphisms, are not only the most abundant, but they're also the best characterized. So there's different kinds of genetic variations, insertions and deletions. There can be whole sections of a gene that are just missing. The glutathione transferase that's involved in vitamin C metabolism, roughly 20% of Europeans and 50% of East Asians are just missing a chunk of that gene, and they're not able to produce a functional protein. Now, it might seem that an insertion or a deletion, where a big chunk of the gene is just missing, is something that can be lethal, and it can be in some cases, but in other times, you just walk around and not even know you have it. So I know myself, I have a deletion in the glutathione transferase T1 gene. I genotyped myself, and I'm just missing that chunk of DNA. It just means, there's been some evolutionary benefits in some instances to having those kinds of deletions. And that's why some of these variations are so common in some populations, right, where we think it's a bad thing, and it can be in some circumstances, but in other circumstances, it's actually a benefit. Does anyone know of an example of a genetic alteration or a mutation that is generally not very good because it causes a disease, but has an evolutionary advantage? I don't know if you guys ever studied this in biology, maybe even high school biology as an example. Anyone heard of sickle cell anemia? So that's caused by a genetic alteration. And do you know what kind of advantage that confers? Obviously it's a disease and it causes adverse effects because blood cells aren't fully mature. Yeah.
It's against malaria or something.
Malaria, yeah. Yeah, they seem to be resistant to malaria. So in certain parts of the world where malaria is endemic, those who have sickle cell tend not to be as impacted by it. So there's other kind of examples like that where, again, some kind of mutation or genetic variant seems to be linked with some adverse outcome, but in other circumstances, it's actually favorable. So something to keep in mind, and this is why really we've been moving away from this concept of calling something mutation or a disease-associated gene. Although in instances, obviously, it does cause a particular disease, it's just not always necessarily bad in all circumstances. I mentioned last week as well that Some genes can be inherited in multiple copies. Remember the example, which gene I mentioned? It's one of the genes that has several copy number variants. That's a nutrition-related gene.
Tasting bitterness?
I know that wasn't the example I gave, and I don't know that bitter taste receptors have copy number variants. They could, but the ones that are characterized so far, they're due to SNPs that cause the bitterness. But you're not too far off, at least anatomically. What gene did I talk about last week that has copy number variants?
Is it salivary amylase?
Amylase. Remember, this could be on your exam. And so we inherit 2 copies of most genes, one from our mother, one from our father, but again, one of the parents can give us multiple copies of a particular gene. So the amylase polymorphism can have, I think, anywhere up to like 10 copies, maybe more. And so you can imagine if you have 10 copies of a gene that's producing a protein, you're getting a lot more protein than someone who just has 2 copies. In some cases, there are SNPs that can be used as tagging SNPs for copy number variants. Has anyone heard of tag SNPs before? You can imagine what the name or the term means. Like you're using a SNP as a tag for something else. So it turns out that some people who have these multiple copy number variants also have a SNP nearby, or a SNP within the gene. And that SNP is a tag for having multiple copies. We actually use that in my lab, because to measure copy number variants is pretty expensive and a little bit more time-consuming, but SNPs are very simple to measure. And so what we did is we looked at this SNP that seems to be inherited with multiple copies. It's in what we call linkage disequilibrium, or LD. So linkage disequilibrium means sometimes whenever you have one copy of a SNP in one region, you almost always get another SNP at the same time, because during crossover, you inherit chunks of DNA together. So sometimes we can use SNPs as a surrogate measure of a copy number variant. So again, even though CNVs, or copy number variants, aren't as numerous as SNPs, because they cover kind of large swaths of DNA, in some instances they can actually be viewed as kind of impacting a broader, what's the word I'm looking for, proportion of the genome can be related to copy number variants. But SNPs are the most abundant, most well characterized. Copy numbers, because there's big, large chunks of DNA that are that can be inherited in multiple copies, it can cover a bigger portion of our genome. So I basically stated this. We inherit 2 copies of most genes, one from each parent, and then they come in different forms. These are just some examples. So the bottom line is, in terms of possible genotypes for at least SNPs, you basically have the homozygous for the what we used to call wild type. Don't really use that term anymore, but you might find it in older papers. So if you're looking through the literature and they talk about wild type, it just means it's the most abundant. And it's actually the most abundant in the population that it was first identified in, because some nucleotides, some genotypes are more abundant in one ethnic group, but they're rare in another ethnic group. So again, we just think of generally now major and minor variant, major and minor allele. So an allele is a strand of DNA that has a particular nucleotide at a given position. Now, sometimes the biological function of a polymorphism depends on whether or not you have one copy or two copies. So if you have one copy and you're a heterozygote, sometimes that substitution can have an effect. In other cases, it doesn't have an effect. You need both copies to have an effect. You guys remember what that's called? Like 2 copies. If you need 2 copies to have a phenotype, what do we call those? Starts with an R. Recessive, right? Something that just a single copy can have it is a dominant effect. So in some cases, biologically, you have to have the homozygous of that Meyer allele to see the phenotype or the manifestation, and that means it has a recessive effect. Sometimes, and it's always nice when we see this, we have what's called a gene dose effect. And that's where the homozygous has a particular response, the heterozygote is intermediate, and then the homozygous for the minor allele is even further. It's like a stepwise effect, gene dose effect, meaning with one copy of that variant, you have a certain decrease in enzyme activity or receptor binding, and in 2 copies, you have even more. And that's also sometimes called an additive effect. Okay, so you have recessive, where you need both copies, dominant, where you just need one, and then additive, where, just as the name implies, with each increasing variant, you get a different effect. Okay, so you all know, of course, we inherit half our genome from our mother, half from our father, and so when it comes to a lot of these traits, we generally view them as coming from directly from the genes that we inherit. And you saw this slide last week. It's just to illustrate a single nucleotide polymorphism. If at a given position, generally we used to use 1% as kind of a cutoff for calling something a SNP, and anything less than that used to be called a mutation. Now the terminology is changing a little bit, and sometimes you might also hear SNV for single nucleotide variant. So SNP or SNV essentially means the same. A single nucleotide is altered. So 3 billion base pairs. A lot of things can happen, a lot of things can go wrong. You get all kinds of SNPs anywhere along that area. And so there's been some interest in trying to categorize the different kinds of SNPs or SNVs in terms of the functional consequence. And so this flowchart just tries to separate the different kinds of SNP. So one question we often ask is, first of all, is it in the coding region or not? In other words, is it going to end up affecting one of the exons? Or is it an intronic SNP? Right? Remember, a gene is made up of exons and introns, and then when that gene gets spliced, we get rid of the introns and the exons come together, and then eventually we code the mRNA from those exons, and the mRNA is used as a template to make proteins. Also in that gene, we have the 5' untranslated region, where promoters usually bind to affect the expression of that gene. Right? And then we have the 3' untranslated region as well. It seems to also impact, it has some regulatory functions as well. So if we focus on the coding region, we can have, depending on the substitution, either a synonymous SNP or a non-synonymous SNP. Does anyone know what that refers to?
A synonym, like the mutation doesn't affect the phenotype, although it affects the gene, but not synonym is
Okay, very close. It doesn't affect the amino acid sequence, because that's what we're looking at first, right? So we don't really know if it affects the phenotype, but at least it doesn't affect the amino acid. Remember from biochemistry, that table where you have your codons, and your 3 nucleotides that make up your codon, right? TAG, AGT, remember those? And remember that some amino acids are made up of 3 different combinations, right? So a CCT and a CCG can all make lysine. That's not true. I'm using that as a kind of hypothetical example. So why is that good? Why is that good that different nucleotides can make the same amino acid? Yeah.
Allows for some redundancy.
Yeah. So if you do get a mutation or if a variant occurs, sometimes it doesn't even matter because you're still going to get the same amino acid. And so it's not likely to alter the biological function of the protein. But in some cases, you have a SNP that is non-synonymous, meaning it does result in amino acid substitution. Now, the different kinds of amino acids varies, right? Because if you have a polar amino acid being replaced by another polar amino acid, might not have as big an effect on the whole protein. But if it's a totally different kind of amino acid, it could have a very significant effect. Now the non-coding region. You can see here, this is a bit of an old slide. It doesn't kind of go any further. It almost implies that it's not really that important. Is that true? If you have a mutation or polymorphism in the non-coding region of a protein, does that matter? Some of you shaking your heads. Why does it matter?
I think that kind of like promoters fine, it's like stability.
Yeah, absolutely. If it's in the 5' N-transit region, affects a promoter, it's not going to affect. I think actually lactase, it's related to polymorphism that's outside the, well, I know it's definitely related to one outside of the coding region. I think it might be in the 5'. Where else might a polymorphism occur that's not in the coding region that can have profound effects on what's produced? So 5', yep, can affect it. 3', same thing, could affect the stability of the mRNA that's produced. What about introns?
Oh, I was gonna say if there's like a stop codon, sort of, that would be.
Yes, stop codons definitely, like a premature stop codon can have profound effects, but those we would consider to be in the coding region because it is that coding, and instead of amino acid, signal is to stop. These are one of the stop codons, and you have what's called a truncated protein that may or may not function normally. Oftentimes, sometimes it will even be lethal. What about polymorphisms in an intronic region?
That's gonna maybe affect the splicing of the mRNA.
Yep, absolutely. So it's if it is affecting the splice site, then those could be actually even brutal, right? Because all of a sudden it's just things not going to splice properly. Either it's going to miss an exon entirely, or it might even result in again a truncated protein where you just have a portion of that protein. Are you going to add to something different to that, or?
Oh, that's my question.
Yeah. Okay. So Yeah, so again, the non-coding ones we used to not pay as much attention to, but really they can be even more significant. So again, the non-coding do not affect the amino acid sequence. The coding can affect it, again, depending on whether or not it's anonymous. And then the amino acid sequence can generally stay the same, or it would change, and then the amino acid changes may affect the protein function or truncate it. So again, in terms of nonsense, that's usually where they tend to be kind of severe changes to the sequence and affecting the protein. All these other ones, again, can affect DNA splicing, transcription, stability, and may or may not affect the protein function. So these are just kind of the basic types of polymorphisms, because again, depending on, let's say, the paper that you're presenting, it might be worth explaining, what kind of polymorphism it is and how it's affecting the response to a dietary factor. So this is just a figure that illustrates missense mutations and nonsense mutations. Don't really need to get into great detail, but you can refer to the figure there, and it shows you if you have a truncated early stop codon. TAG is, I think, a known stop codon, and that's what you can get there. Some of the things to know about SNPs, again, there's been efforts to try to standardize the naming and the nomenclature of SNPs, and we now always use RS numbers. That R should actually be a lowercase r. It's just because it was started from the beginning of the sentence of capitalized. The RS number refers to it's a unique identifier for a particular SNP. So a particular SNP that affects caffeine metabolism is RS762551. I just happen to know that because we published a bunch of papers on it, and that number just sticks in my head. RS762551. You don't have to memorize that. I'm not going to ask you that on the exam. But generally, well-studied polymorphisms, those in the field, know the RS number. It's almost like a phone number. They just kind of know how to say it off the top of their heads. And again, it indicates a very specific location and a very specific gene. So other ways of referring to a polymorphism refers to the nucleotide. So then you have the RS number. So these are 4 different ways of referring to the same polymorphism. And again, it's important to understand this because if you're looking at a paper and you're studying a particular polymorphism, and you come across another paper and you're like, oh, this paper doesn't talk about the RS1801133. This is talking about the 667C to T. Well, that's the same SNP. Okay, it's just older nomenclature and a different designation. So this refers to the nucleotide substitution, and it's a C to T. This refers to the amino acid substitution because in this case it happens to be a what kind of SNP? A coding SNP. Because it's causing an amino acid substitution. Alanine is being substituted for a valine. And so this is the amino acid position. And remember, every 3 nucleotides, there's an amino acid, right? So if you do 222 times 3 is 666. And that position also includes 667. So it's 665, 666, 667. That's the one that makes up that amino acid at this position. You also recall from biochemistry that amino acids can have single letter designations, right? Again, just check your biochemistry. This is another way of doing it. So alanine is just a letter A, valine is the letter B. Does anyone know what amino acid has the letter R? You can make it up because I don't know personally, but I know that R is one of them. Could be arginine, I don't know, because arginine can't be A because alanine has that. Anyhow, maybe someone can look it up. Is it right?
Yeah.
Arginine? Okay. Sure, I don't care. Anyhow, take-home message really is different ways of identifying the same polymorphism. MTHFR is a well-studied enzyme that is involved with folate metabolism. And this polymorphism actually is one of the oldest metabolic polymorphisms that have been identified. And it was discovered by a group at McGill, Dr. Ruby Rosen and her group. Some valuable resources in terms of databases that you can use to look up SNPs are the ones that are listed here. DBSNP is kind of the central repository that is used to catalog these variants, but there's other resources. SNPedia is, some people find that useful because oftentimes if you plug in your RS number, it'll actually give you links to recent papers on that particular polymorphism. So again, just in terms of expanding your research skills, these are useful databases to use. So I mentioned last week kind of nutrigenomics versus nutrigenetics and nutritional genomics. This I like to have kind of as a broader definition, and then when we look at how genes and diet interact, ultimately personalized nutrition is one of our goals. So variations can affect various levels. As I mentioned, we don't just jump right to the protein. There's also the epigenome. So genes get all kinds of epigenetic modifications are possible. Things like methylation of DNA that can alter the biological function. And so what we're going to do today is just kind of go through the different kinds of omics using, when we look at really considering the flow of genomic information and how that translates into a physiological effect and where nutrients interact with the different levels of regulation and how nutrients can actually regulate these different steps as well. So the omics technologies, again, genomics refers to genome changes. Transcriptomics, the expression of the mRNA in a particular cell. So the genome is the same in every cell that has DNA. So not including red blood cells, but every cell has the same genome. But the transcriptome is very tissue specific or cell type specific. So the expression of different genes, that global changes in gene expression is not the same from one cell type to another. And this is important not only for from a research perspective, but even from a, kind of a testing perspective, right? In terms of clinical applications. It's important because I often, I hear this from, sometimes when I'm giving a talk on genetics and someone will say, well, but it depends on whether or not the gene is turned on or turned off. And I'm like, yeah, that's true. But you can't just measure the expression of a gene, because if you do, where are you going to measure that? Are you going to take a muscle biopsy? Are you going to take a liver biopsy? Cheek cells? The expression of various genes are going to be totally different. In fact, one of my collaborators at the University of Laval did a transcriptomic study where they looked at typical cell types. So they did blood, they did adipose, and I think they did muscle biopsy. And they looked at the transcription profile of people just under kind of fasted levels. And they had these kind of three overlapping circles where there are some genes that are similarly expressed in blood as adipose, just a few of them, and some that are commonly expressed between muscle and adipose, and others between muscle and blood. and then even fewer that are similarly expressed in all three, where there's that overlap. And the message was really, you can't extrapolate from one tissue type to another. You have to understand what's happening locally. So that's the transcriptome. Once you have the mRNA levels, obviously we know that generally, the higher the mRNA, the higher the amount of protein is going to be produced. And so we can use proteomics. This is 2D gel electrophoresis. You might have seen a picture of this, where based on molecular weight and charge, proteins have a certain migration pattern on an agarose gel. And based on the pattern, we can look at specific proteins that are different in a control group versus an intervention group. Right? So you can do a study where you feed people, grapefruit juice for a couple of weeks, and then take some tissue biopsy and look at the proteins that are changed and look to see if any pattern looks different. That's one way of proteomics. There's more sophisticated ways now using mass spectrometry. Similar approaches with lipidomics, also to look to see how dietary factors can affect different lipids in a cell type. And then we have the microbiome. So I don't spend a whole lot of time on the microbiome because there's a whole course devoted to that. If any of you are taking Dr. Kamelli's course, NFS 485, it focuses a lot more on the microbiome. But we'll touch on it in a few cases in this course because there is a lot of interest in the microbiome in terms of personalized nutrition. Whether or not, or if we can, use the microbiome to predict response to diet. So, similar to lipidomics, which is actually kind of a branch of metabolomics, but focuses on lipid compounds, some believe that it's most important to look at the metabolites, because those are the ultimate products of metabolism. And we try to, again, using various techniques like NMR, nuclear magnetic resonance, or mass spectrometry, we can use those approaches to measure different kinds of metabolites, again, in a particular biological fluid. So the urinary metabolome is not the same as the blood metabolome or the salivary metabolome. So again, different tissue types and different biological fluids will have different profiles. So it depends on the research question, what you're trying to ask. So there's really two things I always want you to kind of think of. There's the research perspective, and then there's the application. Right? Because a course like this really focuses on applying the knowledge and translating the science. So we use a lot of these tools in a kind of a research setting, and where relevant, we look to see where we might be able to use them in a clinical setting. Right? If we discover that there's a certain metabolomic profile that shows, hey, this is something, there's a disease here, or there's a different type of diet this person should be following, then that's something that we want to refer to. In fact, last week, I think when I touched on inborn errors of metabolism, right? PKU is an example. I'll talk about it a little bit more today. Every child that's born in Ontario gets tested, and even though it's a genetic disorder that's managed by diet, we actually use metabolomics to detect it. Right? So every infant that's born, they take a heel prick blood test, and then they measure the metabolites, just because there's so many different genetic variations that can impact that one enzyme that can result in a very specific metabolic perturbation. And so why try to measure all these possible genetic variants when you could just measure that single metabolite? So again, in terms of some practical applications, sometimes metabolomics is useful. Now these arrows really should be bi-directional, because certainly not only do nutrients impact all of these different levels of regulation, but there's also kind of a bi-directionality. Right? especially when it comes to genomics, right? Because not only can nutrients impact the genome, but genetic variants can affect the way we respond to nutrients. So in terms of some of the basic terminology, again, the genome, basically a full set of DNA in a particular cell or organism. Genomics deals with more of the sequencing, the assembly, the annotation, and understanding the structure and function of genomes. Genetics generally refers to specific genetic alterations, whereas the genome was kind of a broader term. We specifically think of genetics as that study of what we're inheriting. The transcriptome, or transcriptomics, looks at genome-wide gene expression, and again, that is cell type specific. Important to emphasize that. Proteomics is a similar approach, but looking at all of the proteins in a particular cell type or biological fluid. And then metabolites, metabolomics is similar to that. There's another term, metabonomics. So there's metabolomics and metabonomics with an N. So for those of you who are interested in understanding or studying this a little bit further, those are kind of two competing terms that researchers in the field were kind of using around the same time. Some make a bit of a distinction between metabolomics and metabonomics. I can never fully recall the difference. It's not really that critical. Bottom line is it's a similar approach, understanding the metabolites in a particular cell, tissue, or biological system. And then lipidomics is very much the same as metabolomics or metabonomics, except it's focusing on the lipid-soluble compounds. All right, so I'm going to go just through each of the different omics very briefly. And again, these are things that I don't want to kind of read just what's on the slide, but just try to illustrate some examples where it's relevant. I'm just going to animate these in because you have them all in front of you. Again, recognizing that we have 3 million base pairs and that these variations can happen either as SNPs, copy number variants, insertions, deletions. There's also variable number, tandem repeats, other kinds of more obscure types of genetic variations that occur. Yeah, so here's really where it gets relevant to what we're studying here, is recognizing that these the variations in the different genes can obviously impact our response to nutrients and how we metabolize them, our eating behaviors, food intolerances, and all of that. A large chunk of research that's conducted in the field of genomics doesn't really think about nutrition. It's almost like a kind of an afterthought. They're looking and they're trying to discover mutations and genetic alterations that cause diseases. Obviously, there's a lot of interest in understanding how we respond to drugs and toxins, and so those are other branches of genomics, similar to nutrigenomics, where there's obviously greater interest because there's something you can do about it, right? It's obviously important to know how genetic alterations cause disease, but it's equally important, if not even more exciting, to understand how it affects our response to nutrition and what we can do about that. So epigenomics is very much related to genomics, and it also deals with inherited traits. So we used to think that it's just the genetic sequence that gets inherited, but these epigenetic changes can also be inherited. And again, this is very much tissue specific. Right? So a lot of these epigenetic modifications occur in one cell type but not another. Lactose intolerance is a classic example of that. And it also explains why some cell types don't produce certain proteins because of certain epigenetic modifications that occur in that cell type, and it basically silences that. I've mentioned DNA methylation many times as really, one of the best characterized types of epigenetic modifications. There's also histone modifications, and those are other common epigenetic changes that happen. And so there's interest in this field now called nutrient epigenomics, which studies the effect of nutrition on the epigenome. Right? Because we know that some of the regulatory effects that happen by nutrients is by causing epigenetic modifications. And again, it's bidirectional. So just like nutrients can cause epigenetic modifications by donating methyl groups, for example. So the whole folate pathway is involved in producing methyl groups that cause methylation of sequences of the genome. And again, it's bidirectional. So nutrients cause epigenetic changes, and then those epigenetic changes affect the way we respond to nutrients. And again, it just depends on the type of epigenetic change that happens. Diseases can occur or can be altered, the risk can be changed. And again, very important that these epigenetic changes can actually be inherited. And so a classic study is one where a group of researchers got some mice and they fed them 2 different diets to the point where it changed the coat color of these mice. And that's caused by an epigenetic change. And sure enough, these were called agouti mice, that change in fur coat color actually was inherited and passed on to the next generation. And so that's something that's of great interest, of course, because it makes us wonder, can we actually change our appearance or our height or our eye color based on our environment? And if so, can that be inherited? It's still something that's really much in the kind of the research in the side of things. Yeah, so again, some of these changes that can happen, in other words, the maternal diet. So the diet of the mother can affect the embryo by causing epigenetic changes, and that impacts, of course, the offspring, and then it can be inherited as well. This is just a diagram just showing what histone modification looks like. And you can see the DNA as it unravels. It's coiled around these histones. That's how we're able to get DNA to be so tightly packed in our cells. All right, let me pause here, and we'll take a short break, and then we'll come back and carry on for the transcriptomics. This is probably a good time to connect with potential group partners. So if you thought about getting together as a group, it's a good idea to share contact information now. And if you want to, you can send me an email and just put NFS 487 in the subject heading so I don't miss it. And make sure that you copy the other members of your group. And importantly, please make sure to tell me if any member of the group is taking 394 or 494, because that's going to impact which day you're presenting. I've already connected with Dr. Metherell, and so we'll make sure that those students who are taking 394 or 494 will, even if they're presenting that day, they'll be presenting earlier on in that day, and he'll still be able to come to class because he knows that peer evaluation is part of that. So take the time if you want, just a few minutes to either have a bio break or connect